Hi there. My name is Raut. I'm a postdoctoral fellow at the Allen Institute and the University of Washington, Seattle. I'm really excited to talk to you today about our recent work, which you can find at this link here, and which involves this awesome team of collaborators entitled Arousal as a Universal Embedding for Spatiotemporal Brain Dynamics. Now, a goal of this work really is to unify a few different pockets or sectors of neuroscience, and to do so using tools from dynamical systems theory and machine learning. And that's kind of a lot, so I'm going to give a really streamlined kind of overview of this work in this talk. And I'd really encourage you, if you're interested, to check out the paper and also the references throughout that paper that really give you a picture of how this story has unfolded across these different areas of neuroscience. So let's jump right in. What I'm showing you right here is ongoing brain activity recorded from an awake mouse. Now you can imagine just kind of looking down, and this is all measured at the surface of the brain. And what you see is actually very typical of what you see also in the human brain, in mammalian brains in general, vertebrate brains, even invertebrate brains. You find these, in the awake state even, you find these ongoing spontaneous patterns of large-scale brain activity involving the coordinated behavior of thousands, even millions of cells. And naturally, we want to know, where's all this activity coming from, right? Is the mouse looking at things? Does it depend on what the mouse is thinking about? What are all these different brain regions always saying to each other, right? Now, the key idea that we're trying to put forth in this work, and what I'm going to try to elaborate on in this talk, is that ultimately, this might be a lot simpler. A lot of what we see up here may ultimately be reflections or manifestations of some underlying intrinsic regulatory process for the brain that's continuously regulating brain-wide physiology. Not only that, but it's also regulating brain-wide physiology in relation to organism-wide physiology and behavior of the organism. So kind of synchronizing everything into a continuous flow. Now, I'll get into how we actually might test a hypothesis motivated from this perspective, but ultimately where this is going to take us is to this idea that, in theory, if this is on the right track, then all I would need is some other measurement from the organism that is coupled to this process that we're hypothesizing is indeed global. And in theory, that measurement should carry enough information to basically know what's going on in this brain-wide activity. And we're going to actually do this using something as simple as pupil diameter. So just that little black dot in your eye, mice have this as well, and it's continuously fluctuating up and down. And what we're going to show is that that carries actually enough information to be able to get pretty good reconstructions of this large-scale brain activity that you see across the mouse brain. Now, before we get to that, we're just going to back up, and I'll give a little overview of brain dynamics in general. I know a lot of people interested in dynamical systems theory watch this channel. And so the upshot really is that I believe that this perspective is very valid, very useful for looking at organisms and thinking about their brain activity. So organisms and their brains are intrinsically dynamic, and they really pose a challenge when we go towards modeling these dynamical systems for the usual reasons, right? They are very high dimensional. A really limiting challenge that we have in biology and neuroscience is that we can only measure a tiny subset of the degrees of freedom of the system, either a particular spatiotemporal scale or just a few of the relevant biological elements, cells, etc. And hidden in all those unmeasured scales that are a lot of feedback interactions, nonlinearity, circular causality, right? So we can only access a tiny subset of those variables at one time, and there's a lot of craziness happening in the background. And so you might think, okay, well, what do we do now, right? This seems like an impossible task, but I think nature has given us a path forward, and that is to realize at the end of the day, organisms work, right? Brains work somehow in spite of all this. And so what that means to me, I think, is that this high-dimensional configuration space of the organism. Instead of exploring all of that, instead of every cell doing its own thing, right, there's some low-dimensional behavior that the system exhibits, right? There's some level of coordination, pieces working together. And so I think as a scientist, as a modeler, our goal can then become, or at least my goal, is to uncover those intrinsic constraints and use them to our advantage to relate the high-dimensional data and observations that we are met with, relate those to some underlying organism-intrinsic low-dimensional processes, right? And the starting point for this is going to be what I'm referring to as arousal, what different fields have referred to as arousal, going through neuroscience, biology, psychology, ethology. You can think of this as basically, from the data-driven perspective, principal component number one. And that kind of shows up in all of our different measurements as really a dominant component, especially on this time scale of seconds. So what I'm showing you here is a very beautiful paper and very well known, especially in neuroscience, from a few years back that basically showed that in an awake mouse, on the time scale seconds, you have these ongoing fluctuations that are coordinated across the locomotion behavior of the animal, pupil diameter, whisking behavior, this is a mouse. All of these are tightly linked to ongoing fluctuations in population level neural activity. So again, thousands of neurons all kind of behaving in this coordinated fashion in relation to behavior. But it's not just this set of observables, okay? In different pockets of neuroscience, you also see that, so for example, over here, that the ionic environment that these neurons are bathed in, properties, the chemical composition of that environment is continuously changing on this time scale of seconds. You also see this for different cell types in the brain, like astrocytes, and the metabolic properties of the brain. Again, all fluctuating on this time scale of seconds. And these fluctuations occur even at the earliest stages of sensory input. So even at the level of the retina, already you're starting to see information coming from the world being shaped according to these ongoing fluctuations in arousal. But what exactly is arousal, right? We can see that it's probably important to get a handle on this as it's popping up everywhere. But it's kind of abstract at this stage, and we don't really know how to study it. So to make things concrete, how we're going to study it in this work here is we're going to start off with a typical set of measurements. So here, this is an awake mouse, and we measured something like pupil diameter, whisker motion of the animal, and then brain activity. These are just a couple example time traces. And what you can see here right away is you see these coordinated fluctuations across these different time series. And naturally, one way to interpret this might be to say, okay, every time the mouse whisks, right, it's causing these changes in pupil diameter, it's causing these changes in brain activity. That leaves unaddressed this question of what's actually causing the mouse to whisk in the first place, right? What's causing the mouse to move its whiskers in the first place? And again, if we try to interpret specific causal interactions between these variables, we tend to leave out what I was showing on the previous slide, which is all these other variables that are ultimately coupled to what we've been calling arousal. So the tack we're going to take instead in this study and the perspective we're putting forth is the idea that this ultimately originates from some underlying process intrinsic to the organism. So there are some intrinsic laws of motion that keep this so-called arousal process continuing to fluctuate on the time scale of seconds, even as you're listening to this talk right now, hopefully, or on and off. And as I'm talking right now, you have these ongoing fluctuations. And the idea is to treat these as simply measurements of this underlying process. They're all just related to this underlying arousal process via some functions that we might be able to learn from data. Okay, and so over here we have this organism intrinsic manifold, this low dimensional structure on which these arousal trajectories unfold, and you have stochastic perturbations from inside and outside the organism. But so now to actually work with this kind of data and to test hypotheses, we teamed up with these awesome experimentalists right here from Washington University in St. Louis. And what we're going to do is to measure simultaneously large-scale brain activity and whisker motion and pupil diameter. And ultimately, we're going to try to, from data, go map back to this hypothesized underlying process. Can we link these things back to an underlying process? So to set up this problem, again, we're going to start with this idea that you have some underlying process that we do not observe, and we have access to, through some idealized observation functions, we have access to pupil diameter over here, and down there we have brain-wide activity, or large-scale cortex-wide activity, technically. And our goal is going to be to relate these two via some common state, some common process. So this diagram will be familiar to a lot of people here, this idea of an encoder and a decoder and a latent space in the middle that really contains information about both of these things, right? So this is the type of thing that should be possible if we're on the right track. But there's a catch here, which is, well, first of all, before I get into that, let's talk about why we might actually think that we have brain-wide activity, large-scale brain activity, coupled to this process. Now, this goes back to a paper from a couple of years ago where we looked at, this is now humans, and we found that ongoing fluctuations in arousal of the kind I've been describing so far, so measured with pupil diameter, breathing rate, this sort of thing, are coupled to these large-scale traveling waves that move throughout the cerebral cortex and are topographically parallel to, or respect the functional anatomy that we know about in a way that matches what we see going on in the thalamus, in the striatum, in the cerebellum. So all these major pieces of the brain have these synchronized traveling waves that are flowing in synchrony with ongoing fluctuations in arousal. So, okay, so this is why we are expecting in the mouse that ultimately large-scale brain activity can be thought of as simply another manifestation, another reflection, another function of this underlying process. But we still have this challenge of how do we actually go from a scalar variable over here, scalar observable, to a high-dimensional observable over there, right? And how we're going to do that now is we're going to take motivation, take inspiration from Tockin's theorem, from dynamical systems theory, and what's been referred to as a time delay embedding as the strategy that we're actually going to use here. So we're going to take, so we're going to go to this toy system, this Lorenz system, a popular toy nonlinear dynamical system, simple model for chaos, and it involves these three state variables, x, y, z, who interact with one another through some known governing equations, some deterministic equations, okay? And so it traces out these trajectories over time, etc. And let's say that we access two measurements from the system, and they're just going to be the state variables x and y. So these are two scalars measured from this three-dimensional system. And ideally, we are going to be able to say something, we'd like to be able to say something about the system from which these variables came from, right? So we want to be able to say that they came from the same system, that the system has these properties, there's three dimensions, etc. And so the question is, how do you actually go about doing that? It turns out that because, let's start with x for now, because the motion of x, the time evolution of x, is entirely determined by the interaction between x and the other variables in the system, y and z, that are all related through this known governing equation, it turns out that x and the time history of x are sufficient to encode that information and basically reconstruct the underlying attractor from which it came. So here what I'm showing you is x plotted against a couple different lagged versions of itself, and you see that basically you end up with a structure that looks quite similar to the original one. Now it looks kind of stretched and warped in different ways, and so the geometry is not necessarily preserved, but ultimately the topological information from the original three-dimensional attractor is preserved in this reconstruction, what's been referred to as a shadow manifold for the original system. And you can do the same thing with y over here. And ultimately, you end up with these different embeddings, different views of really the same system that can be interrelated through different maps, different functions. Technically, these are diffeomorphisms. But basically, to make this come alive a bit, I guess, to get this point across. So you can imagine you have this state unfolding down here with this green dot. And the idea is that this motion can be viewed in different ways, right? You can have a time series snapshot of it over here, a time series over here, and you have the motion in the delay embedded spaces for X and Y over there. The idea is that these are all just representations, different representations of the same underlying process. There's one process going on here, okay? And that's exactly the perspective that we're trying to take with us when we approach the brain activity. The idea that ultimately you have one arousal process going on. And the evolution, the time evolution of this process, we are observing that in the pupil diameter, in the large-scale brain activity. In order to test this hypothesis, we're going to time delay embed our pupil diameter into this high-dimensional delay space that theoretically uncovers this missing information, the information that we lost when we went from this arousal manifold to this scalar measurement, pupil diameter. And once we have that, recovered that information, we can probably collapse this down a bit more to some low-dimensional manifold that preserves everything we need from this hypothesized arousal manifold. And ideally, we can learn a mapping from that space, a time-invariant mapping, now that we've unfolded that attractor in the space. Time-invariant mapping that says for each point in time, as a function of my position on this arousal manifold, what does my brain activity look like at that point in time, as measured through this wide-field optical imaging that we have over here. So now that we've set all this up, we can go back to what we set up at the start of the talk, which is this video showing on the left the original brain data, and on the right, this is a prediction in held-out data where we're using the pupil diameter in that held-out data, and we're using our mappings that we learned in the training data to predict or reconstruct what brain activity should look like at each point in time. So I'll let this play out a little bit, and you'll notice that, okay, they're not perfect mirror images of one another, but I think the take-home point, what I'm hoping to convince you of, is that it becomes hard to really think about what's going on over here as something entirely different from what we see over there, which is ultimately all information that is just contained within ongoing fluctuations in pupil diameter. Okay, so the goal of this talk isn't just to basically say that you can do this, but to give us an explanation for why we are able to do this, and to give some background for how we arrived at this prediction. So, if we look to quantify this, I'll just go through this part of the talk pretty quickly. We do quite well, in fact, even with linear regression and just pupil diameter with no delay embedding. And that's because that first principal component explains a lot of the data that we see, a lot of the variance. But the key point is that the delay embedding and the nonlinearities allow us to capture that kind of continuous flow of the spatiotemporal dynamics that we see in brain activity. And this is also kind of clarified in the mean subtracted data, where you're taking out basically that proportion of the variance that just kind of represents going up and down in activity. And we see that if you take that out, you're still explaining quite a lot of the variance in this data, the spatially specific information that's not globally represented. So I'll show this real quick just to build some intuition for how we're thinking about this and what the data looks like. So another way to think about this is each point in time you can assign them, and you do some clustering and say, hey, you look kind of like this brain state, right? This spatial pattern. You can go through and do that for the entire set of image frames. And you can visualize now the entire brain recording as a trajectory in this three-dimensional space. And now the axes are defined in terms of the leading principal components of the brain activity. And what you can see is that different colors cluster to different spots on this manifold of this state space, right? And so the idea is that you have a characteristic flow, characteristic dynamic that goes through these different states. Now, if we take those same colors and plot them on a trajectory that's reconstructed in the delay coordinates from pupil diameter, we see that once again, in fact, the colors segregate out to different spots of the state space. So what this is getting at is that this spatially specific information represented by the segregation of colors along this state space, where colors correspond to different brain maps, that information is actually, a lot of this is basically there already in pupil diameter. The pupil dynamics kind of recapitulate the spatially specific information. So finally, we'll take this out to a big picture. Okay, this was a specific prediction about wide field calcium measurements, and that we should be able to reconstruct those. But we have this intermediate thing that we've constructed, this so-called arousal manifold, which is intrinsic to the organism, right? So it's not dependent on any observation space. And this is really where we start thinking about this idea of a universal embedding and how we might be able to put together these different pieces of the organism that we typically study in biology and neuroscience independently of one another. So here's kind of the big summary picture. So you have something like this idealized arousal manifold. Here I'm just parameterizing it according to two coordinates, we'll call it Z1 and Z2. And the idea is that you have some basic flow on this manifold. So this is kind of this canonical cycle, if you will, of the arousal dynamics. There's a vector field telling you how you tend to move on this manifold. You can also learn functions that take you to observables. So again, these are recorded at Washington University in St. Louis. We have the wide field calcium images that tell us basically, on average, what to expect at each piece of this manifold. What does the wide field calcium look like on average? What can I expect to see there? But now we can go through and use neural networks or use linear regression, use whatever else, to basically learn functions that take us to different measurements from this same latent manifold. So recorded simultaneously with the calcium measurements were measurements of approximating oxidative metabolism in the brain, of blood flow, hemodynamics. And so we can kind of plot those on the same space and say, hey, for each spot on the manifold, what can I expect to look like from each of these different observations? And then, of course, we can throw in here the scalar observables, pupil diameter, and whisker movement. Now, a key point is that while these were all recorded simultaneously, the idea is if we have this organism intrinsic manifold, we can now go and use this to get kind of a group level or averaged picture of what this tends to look like across mice, even using observables that were recorded separately in different sessions, different data sets, different experiments. So now what we're going to do is turn to the Allen Brain Institute, who has released publicly available data, really exceptional data using a set of modalities that is very complementary to what was recorded in this data set over here. And so behavioral variables, physiological variables like pupil diameter and running speed, we can play the same game over there using functions that we've learned in that data. And we plot them on the same manifold using transfer learning, which I guess is discussed a bit more in the paper. But the key point is you can go through and now do this for another, more observables, basically, that are served as the basis for entire sectors of neuroscience. A lot of amazing work looking at different, in this case, phenomena related to the state of the hippocampus, measurements of basically other electrophysiological measurements of the brain activity. We can start to think about all of these things as resulting from functions of this unified process. So in sum, you have three things. You have this latent manifold, you have function taking you to the flow along this manifold, and then you have observation functions that take you to an expectation of what that looks like from the perspective of each of these measurements. So in sum, preprint is available at this link, and the real take-home point here is that when we see these amazing spatiotemporal patterns of brain activity, the idea we're putting forth here is that ultimately this might come down to a manifestation or reflections of some underlying organism-wide process that is regulating everything going on in the organism and is not actually specific to the brain activity. So funding sources are listed over there, and big thank you to the groups that made this work possible, in particular the Shanahan Family Foundation and Allen Institute, which has funded a lot of me being able to do this work. So please reach out. Email address is over there. If you have any questions, would like to chat more, any complaints, I'd love to hear from you. So thanks so much for watching.